influence species diversity. So we know that species richness of an area increases with the size of the area and decreases with isolation. So this first graph, we're seeing how as island size increases, so does species richness. Whereas when the distance the island is from other similar areas, as it is further away, species richness is lower. If that island is closer to a similar area, the species richness is higher. So here what we're looking at is for a given area, islands near New Guinea support more bird species than islands further away from New Guinea. So the near islands have higher species richness, the far islands have lower species richness. And so as ecologists were seeing this trend in the number of species concentrated in an area in response to Hutchison's paper on why are there so many different kinds of animals, a lot of people were starting to combine the studies of evolutionary biology, population biology, as well as population ecology, all of this during this history, or the time in history in biology we refer to as the modern synthesis. The modern synthesis is where Mendelian genetics and Darwinian evolution came together, and the study of both primarily evolutionary biology, but as it started to develop as its own science, ecology became more and more quantitative. And this is one of the key stories in that transition is the development of the equilibrium model of island biogeography. And I gotta admit, this is one of my favorite parts of community ecology, where it mixes theoretical ecology with stuff we really see on the ground. It's not just an idea, it is a mathematical theory that was based on observation and has been demonstrated to be correct numerous times over and over again. And it really shaped the development of ecology in the 1960s into the 70s, and it started to trail off in the 1980s as this theory was demonstrated to be correct. But as we're going to see, this became the foundation of conservation biology. So, species richness on islands is theoretically a balance between immigration and extinction rates of species. The rate of immigration is the rate of arrival of new species to the island. And you can anthropomorphize this. It is easier to immigrate to a large island that is close to the source of potential colonists. Okay? In other words, not isolated. The rate of extinction is the rate at which species went extinct on the island. Again, anthropomorphizing it, it's easier to survive, i.e. not go extinct, on a larger island with fewer species because competition will be lower. So this was set forth by Robert MacArthur, who you learned some about in Chapter 1, and E.O. Wilson, who is what you hear many, many biologists of all walks of the discipline refer to as the modern day Darwin. And this was put forth first off in 63 and later again published in a book format in 1967. And what the model demonstrates, it explained the variation in the number of species on islands by the influence of isolation and area on rates of immigration and extinction. So notice we're combining four things here: the isolation of the island, the size or the area of the island, with two measures that typically are discussed more in evolutionary biology: rates of immigration, rates of extinction. Those of you that have already had evolutionary biology, you've done this before. Those of you who will take it in coming semesters, you're going to be ready for it because it is always taught in both ecology and evolutionary biology. So the model predicts higher rates of immigration to islands nearer a source of colonists, and it predicts higher rates of extinction on small islands. Down here towards the bottom, the model explains then that there is going to be a lower number of species on small isolated islands, as well as the model is going to account for a high number of species on large near islands. So we're seeing this play between immigration and extinction. And your first pair of isoclines here, we have immigration to near islands, extinction on small islands, setting forth an equilibrium point where those two curves cross that is looking at the higher number of species that could be present on large islands near a source of colonists. Looking at this other pair of isoclines, immigration to far islands, extinction on large islands, again explaining that when an island is further away, you're going to have lower amounts of species richness. And don't worry, we are going to work through several examples examining these isoclines and their point of equilibrium, which is where the two isoclines cross, and that is going to be your point of stable species richness, or the number of species that would be present on that island, with this interplay of island area, isolation, and the influence those two have on rates of immigration and rates of extinction. It's really not as hard as it sounds. And this is at one point that we need to pay tribute to Robert MacArthur. And this is the eulogy that was written for Robert MacArthur. He unfortunately passed away at the age of 42. He was a very heavy smoker, as were a whole lot of people at that time. And he died of lung cancer at a very, very young age. But this is a person who modern evolutionary biology and modern ecology would not be the way that it is today without the work of Robert MacArthur. So, summarizing the equilibrium model of island biogeography. Island diversity, then again, is the outcome of highly dynamic balance between immigration and extinction, and the rates of immigration and extinction are determined mainly by the area and the isolation of the islands. So, as I was already alluding to, this next wave, after MacArthur and Wilson proposed this theoretical model based on data that they had collected, then you had a whole wave of ecologists in the late 60s and throughout the 1970s whose dissertations were based on testing this model. And this is when community ecology really, really took off. And we started to look at patterns of species richness and species diversity, both in terms of how do you explain those patterns within the area that you're studying, your own habitat or your own ecosystem that you're interested in, as well as global patterns. So really, really neat thing. And of course, I'm getting excited because this is part of my field of research. So species composition on islands changes over time, and this is called species turnover. And Jared Diamond was interested in studying this, that you can have an equilibrium number of species over time, but the composition, the species represented in that number, is going to fluctuate. In this case, with bird species, he's looking at the turnover in the air in the islands off of the southernmost coast of California, and you see that it's this balance of extinction and immigration. And for the majority of these islands, for the period that he was examining, based on records of these species on these islands over time throughout a 51-year period, that's not too bad for written records by amateur ornithologists, seeing that there is this balance between extinction rate and immigration rate. In other words, those kind of salmon or pinkish color bars for the majority of the islands are very close in size to the purple bars, so that there is this balance in this somewhat, if you will, stable number of species on these islands over time, even though the composition of the bird community was fluctuating. Then you have what I love is Dan Simberloff's work. He came to the University of Tennessee where I got my PhD towards the end of my years at UT. And Dan is just awesome. Dan's not afraid to try anything. And he worked with E.O. Wilson. I should also mention that Jared Diamond worked with E.O. Wilson as their advisors on their PhD programs. So you're kind of seeing that Wilson was the one that carried this thread from Hutchison's very quantitative mathematical models through to empirical tests of those models on the landscape. So what Dan Simberloff did is working in the Florida Keys. He wanted to see if if you could basically remove an insect community, would it recover over time to this point of equilibrium based on the area of the island as well as the amount of isolation? of the island. So yes, this man went out there and covered islands and fumigated them to kill off all the insects. They took the cover off of them and they waited over time to see how the thing recovered. So, and you'll see, look how quickly it recovered. Look at that x-axis. We're less than a year there. So the number of species on the near island soon equaled pre-defaunation, I can never say that, pre-defaunation levels, while the number of species on the far island, it took longer. It was still below that original level even after a year. So the blue dotted line represents the number of species that were present on the island before they fumigated it for the near island, and the red dotted line is for the far island, again before fumigation. So it's pretty fascinating to me how the near island, of course, you have a higher rate of immigration, that you had this faster recovery, again supporting the theory of island biogeography, and the far island also supporting it, that it was going to take longer because of the increased isolation of the island. So that's focusing in on isolation, one of our two components there involved in this model. Dan wasn't satisfied with that. Dan then wanted to play with a backhoe. Anyone who's ever been down to the Florida Keys, the water is incredibly shallow around some of these islands. So shallow that you can take a simple raft out to it. In many of the islands, you can actually walk to and carry your equipment. So what he did in the 1970s was to literally reduce the size of two islands. Okay? So he got in there and reduced the size of the island, let it recover to see if there was a corresponding decrease in the species richness of that island. Again, the number of insect species to the number of arthropod species. So the island was, and then the control here was left alone. Notice how the control number, the species richness actually went up a little bit between the two periods. But in both cases, when he got in there and reduced the island size and allowed the island to recover, the number of species decreased with the decrease in area. So Simberloff's work tested both isolation as well as area. Where all this has its practical application is in conservation biology, and that's what this wonderful quote back from the late 80s from E.O. Wilson and Hutchison were emphasizing, is they were emphasizing that thanks to the theory of island biogeography, now what we learn from this mathematical model can be used to plan recovery efforts for not only preserves for species, setting land aside, how big should the land area be, where should it be in reference or in relationship to other similar patches of the same habitat type, as well as just what am I trying to say? Recovery efforts. So it's used in conservation biology both for the preservation of species richness as well as the recovery of species richness in these areas. And so it really brought Hutchison's 1959 paper on the homage to Santa Rosalia, or why are there so many kinds of animals, to full circle by being able to explain why there are so many kinds of species in an area based on the measures of the theory of island biogeography. So from that, people were also starting to expand out from island areas to large-scale global patterns of species richness and species diversity. And this brought in the terminology of biology hotspots, that a hotspot contains a great diversity of endemic species. Those really key central areas of biodiversity only occupy less than 1.5% of Earth's surface, but they house 1/3 of all species of plants and vertebrates. So I've always defined these as if funds are incredibly limited and you have to choose between two areas to put under preservation, if one of them is a hotspot, that's where the money goes. So these started to beg the question again, why is so much of Earth's biodiversity concentrated in these equatorial regions? Notice how they're dead smack in between the Tropic of Capricorn and the Tropic of Cancer. They're right there along equatorial latitudes. And so This has been demonstrated. I only pulled out one graph from the chapter over and over and over with different kinds of species. I think there is one exception to the rule that's presented in the chapter. Mother Nature always has to keep us on our toes. So the number of plant species decreases as you move from the equator or 0 degrees latitude towards the pole, heading up towards 60 degrees latitude. And either way you go, towards the North Pole or towards the South Pole, you see this decrease in the number of species. So late 80s, heading into the early 90s, James Brown, and yes, he got teased for his name all the time, put forth this big paper proposing 6 categories of explanations for why you have this concentration of species richness and diversity in equatorial regions. So, time since perturbation. The tropics are older and less disturbed. More time for speciation, less frequent disturbance reduces extinction rate. This is natural disturbance, not anthropogenic disturbance. Productivity, something we've learned a lot about, that the high productivity, tons and tons of biomass, means more energy to support more species and more individuals. Environmental heterogeneity that we defined in our previous lecture. Lots of variation in the environment, thus more potential habitat areas and niches. Favorableness. Tropical climate has no extremes to limit biodiversity. It's summer all year round. So there is easy adaptation, if you will, to those environmental conditions that allows for that niche differentiation. Niche breadths and interspecific interactions. This brings in the role of biotic and abiotic factors in the environment. This one is so heavy on the evolutionary biology. I'm not gonna hit you hard on this one, I promise, because some of you have not had the class. And then lastly, differences in speciation and extinction rates. This I do expect you to know from evolutionary biology. High rates of speciation, low rates of extinction in these tropical areas leads to great amounts of species diversity. So, other reasons why areas occupied by the biome and latitudinal gradients of species richness, there's a lot of land area in that belt around the tropics. Therefore, we can bring in that concept of the area of the land just on a larger scale than an island. Continental area and species richness again, and then historical and regional influences in the area. So, this I'll let you read more about in the chapter. We'll discuss them in class. And, so what this lecture really wraps down to, why do we have biodiversity hotspots concentrated in tropical and Mediterranean climates, bringing all this information together? It comes down to these interactions of spatial and temporal factors. You have part of species richness and diversity defined by the size of an area and where it's located in terms of closeness to a source or just the amount of area, as well as temporal factors that bring in those historical influences, as well as the flux of both individuals with our rates of immigration and extinction, being species level, as well as species turnover, as not Simberloff, as Jared Diamond did in his research. So we can summarize all this again with these interactions of spatial and temporal factors that influence the amount of biodiversity in an area.